Continue to the last part. In the last part, you know about the plasma membrane structure, and now we want to know about the plasma membrane function. The plasma membrane includes the phospholipid bilayer and some integral and peripheral proteins and also cholesterol and carbohydrate that with this component have different function. This function include communication that in the cell membrane, in the outer side of it, contains some receptors that recognize and respond to the signal molecule that in next part I explain for you about the cell signal. And also the main function of the plasma membrane is a barrier and selective permeability. Physical barrier, the phospholipid bilayer, separate the substance inside and outside of the cell and regulate the entry and exit of the molecules, ion, nutrient, waste materials between the cells. That now we want to know about this main function. Membrane transport divided in two types. Passive process in slide 3 number and active process in other slide. Passive process don't need to energy, and without the using of the ATP, the membrane transport occurred in the cell membrane. This mechanism is for some molecules or ions that transport from the high concentration to low concentration. That this process named diffusion or facilitated diffusion. Diffusion means the materials or ion in passive transport from high concentration to low concentration. But some materials such as the blood gases diffused by facilitated diffusion between the cell membrane. And also the transport of the water done by one transmembrane protein that named aquaporin that transport water from the outside to inside or to opposite direction. But these proteins that most of them are transmembrane proteins, or no, maybe in simple diffusion, they pass from the phospholipid bilayer, such as some hormones, such as steroid hormones, that biochemically, they biochemical structures include the lipid, can pass from simple diffusion. But some of them pass from the transmembrane proteins that act as a channel or act as a carrier. But these channel and carrier, in this channel and carrier, the material transport from high concentration to low concentration. The passive process don't need to ATP, but the active process need to ATP. That the ions or materials pass from the outside to inside or in against direction. In this, in the active transport, maybe mostly involve the transmembrane protein involved in this transportation that carry the materials from outside to inside. When two material or two ion pass from the one part to other part together, this named symport. But if this is a one material, this is a unipole. But sometimes two material pass in two opposite direction. One of them in one part to other part and another part to, for example, inside to outside. This pass active process named the antipole. The two material transport into a different direction. But most of the membrane transportation occur by vesicular process. In this process, some vesicles formed and the materials exit from the cell by exocytosis or enter to the cell by endocytosis. That endocytosis again divided to phagocytosis, pinocytosis, and receptor-mediated endocytosis. Endocytosis is a process that moves the large material into the cell, such as particles, organisms, and large molecules. As you see here, the large material or molecules enter to the cell. When the solid materials enter to the cells, this process or this type of endocytosis named phagocytosis that starts with the surrounding of the material by cell membrane, the edge of the cell membrane meet or fuse together, and they form the vesicle, and this vesicle finally fuse with lysosomes and form the phagosome. Phagosome is a vesicle that includes solid material such as microorganisms or bacteria. Phagocytosis occurs generally in white blood cells such as macrophage and neutrophil because they involve in the remove and digestion of the microorganisms and the debris. The other type of endocytosis is pinocytosis. In pinocytosis, soluble materials enter to the cell. In this process also, as same as to phagocytosis, the cell membrane invaginate, the edge of them fuse together and pitch in, and finally form the vesicles that include the soluble materials, create the pinocytotic vesicles that enclosing the content, and finally they fuse with lysosome and digest with lysosome. But there is one process in transfer of pinocytotic vesicles that named transcytosis. In this process, the soluble materials enter to the cell, form the pinocytotic vesicles, but without fusion them to lysosome, they transfer and exit from other side of the cells. This transportation named transcytosis, that the material transfer from one side of the cell to other side of the cell. Other type of endocytosis is a receptor-mediated endocytosis that is general in most of the cells. In this process, the ligand or some materials join to the receptors that located in the cell membrane. The joining of the receptors and ligands formed a pit in the cell membrane. There are one protein in the cell that named clathrin that generally located in most of the cell, that this protein facilitates the invagination and pitching of the cell membrane and formation the vesicle. Receptor, ligand join to receptors, clathrin proteins aggregate under the cell membrane, facilitate the invagination of the cell membrane, form the coated pit, and finally the edge of them fuse together, pitching, and form the coated vesicles that coat by clathrin. This coated vesicle fusion with lysosomes or endosome and digest it, and finally degrade by lysosome. But the clathrin and also the receptors again recycle to the cell and cell membrane and used for another process. You see here the electron microscope of these processes in this slide. Recently, another protein discovered by scientists as caveolin that's not general in all of the cells. The collagen generally located in all of the cells, but caveolin discovered in the endothelial cells and also in the smooth muscle cells. That again here the ligand joined to its receptors, the caveolin protein accumulate under the cell membrane, invaginate the cell, pitch it, and form a coated vesicle with caveolin. These structures that the pitching done in the cell membrane, these structures named caveola, and finally these coated vesicle transfer and fuse with the lysosome and lysis by them. The caveolin, as I said for you, located in the endothelial cells and also in the smooth muscle cell. The other process or transmembrane transport process is exocytosis. That in this process, the materials exit from the cell. Vesicles form as secretory vesicles in the RER or maybe Golgi apparatus. The vesicle fuse to cell membrane and finally release the products to outside of the cells, and the cell membrane again reversed by endocytosis. Exocytosis have two pathways in the cells: regulated secretion and constitutive secretion. In regulated secretion, one signal such as hormone or neurotransmitter attached to its receptors, these signal regulated the membrane fusion and the secretory vesicle released from the cell membrane. But in the constitutive secretion or unregulated membrane fusion, some materials that produce in the cell synthesis, for example, soluble proteins such as collagen protein, must to exit from the cell. They don't need to signal. These newly synthesis protein or lipid must to transport and exit from the cell. This pathway named constitutive secretion that mostly occurred in the fibroblasts for the exocytosis of the collagen protein. By endocytosis and exocytosis, the cell membrane recycle in the cell. In the endocytosis, the ligand receptors clathrin, the cell membrane enter to the cell, fuse with the lysosome, and after that, the clathrin and also the receptors and also the cell membrane come back or return to the cell membrane. This recycling of the cell membrane by endocytosis and exocytosis named membrane trafficking, that cell membranes come in and exit and return to the cell membrane. Briefly in this part, you know that the cell membrane controls the movement of the substance in and out of the cell, and it acts as a selective barrier and have selective permeability to ion and some organic molecules that control the entrance and existence of the materials inside or outside of the cell. You can study this part from chapter 2 of the Jankuira basic histology.